Well, thank you very much. It's a great pleasure to be here. The purpose of today, I think, and I'm relatively new to this whole topic, but the purpose of today, I think, is to bring my world into your world, which is what's behind this title. My world is from DNA to genomes, and it goes into genetics and disease, but your world is Ring 20. We've not really met before. We've had a few conversations on the phone, we've written a document or two, and we don't really know where we go from here. But we need your help, and hopefully you will benefit from ours as we go on this journey where we start to bring my world into the world of Ring 20 and just see what we can do. So today I'm going to talk mostly about my world, but we will sort of focus the discussions on how it might help and all the gaps there are to understand together. And if there's one sort of watchword of all this, what my world brings is hopefully a new lens or new microscope or whatever it is, a new way of looking at Ring 20 that hasn't happened before. So I'm not surprised you haven't got a lot of answers yet, but what I'm doing has only been developed in the last 10 years, 15 years. So in that sense, it's inevitable. We're only just on the beginning of this new journey. So let me start off. I guess any of these buttons will do, won't it? Yes, there we go. That's a quick overview. I'm going to talk a bit about DNA and the genome itself, what it means, what that term means, and then talk a bit about how the genome applies to medicine. So I'm not going to talk in a great detail, but just give you a bit of a flavour. It's one thing to sequence the genome, it's another thing to look at what is medically relevant from it. And then, of course, to focus into Ring 20. What has the DNA story done for Ring 20 so far? I won't go into a lot about it. But I will then, that provides the background to tell you what we hope is going to happen next and the future prospects and how we have to reorient a little bit to a new problem. So that's what I'm going to talk about. So let me start off with what a genome looks like. And some of you may be more familiar with these sorts of views than many people actually that I talk to. But I thought I would start off at a level that you may be familiar with. This is a genome. This is a microscopic photograph of a genome. These are the chromosomes. For those of you who do know about chromosomes, these are quite simply staining bodies. That's what chromosome means. You stick a stain on it, you look under a microscope, and if you see it, it's a staining body. It's a chromosome. So these are chromosomes. That is an entire human genome on the slide that you can see there. And you can see there just two red dots. We'll come back to the coloured dots in a minute. But the point is there are two red dots indicating that's chromosome 6. You should recognise that. It's chromosome 6. But the point is there are two of them. And why are there two of them? It's because one of them comes from mum and one of them comes from dad. Equal contribution. There are pairs of every chromosome. There is a pair of chromosome 20, pair of 21, and there's either a pair of X's if you're a female or an X and a Y if you're a male. So that's what a genome looks like. So let's go in a little bit more detail because we can't see any DNA yet. So where's the DNA? Well, this is when you start to go into higher magnification. This is into electron microscopy now, which is the highest magnification you can see. And you start to see these strings of beads. And that's what happens when those chromosomes have been teased apart, just with a bit of salt. And if you go at higher resolution still, you start to see, you can see on the left-hand side, that's the same as I'm looking at, isn't it? The left-hand side is a sort of skeleton of one chromosome that was there on the top left. But you see these enormous strings, suddenly these loops of string folding out, and that is DNA. And that's the DNA that is packed in the chromosomes that is part of the genome. And that DNA is what interests us. Now we've got to the limits of microscopic resolution, so everything else is built from models, built from imaging, or is a figment of my imagination. This is a model of the structure of DNA, the double helix, so-called. Very famous discovery. The model was built here in Cambridge, here down the road in Cambridge. It's a very Cambridge story, the whole history of DNA and genomes. And essentially, although you don't see it, but you can sort of see a ladder. I hope you can see a ladder there in that. And each rung of the ladder is one piece or one byte of genetic information. Chemically, what's encoded in all those atoms, which are carbons and hydrogens and oxygens and nitrogens and a bit of phosphorus, what's encoded at each step in the ladder is one base. It's an A or a C or a G or a T, And that's the 4-letter alphabet that makes up DNA and makes up all the information for all of you. There are 3, I'll come to numbers in a minute actually. So that's the nature of what we're trying to do. We can see the chromosomes. We can picture the DNA for those of you who get the chance to look at these microscopes. But inside all this is a thread, a strand of DNA, which is a sequence of letters. Hence the term sequence or sequencing, because we determine the order of the letters. And just like any book, any novel, the order of the letters makes up the words, makes up the sentences, and makes up the meaning and the whole story. And what we need to do is to come back to find out which words in genetic diseases have gone wrong. They've been misspelt. There have been mistakes and errors, and that's what we're trying to do for Ring 20, and that's what we're trying to do for everything else. So that's the code, the genetic code, the 4-letter code, the alphabet, sometimes called the alphabet of life, but that's what we're trying to look at. So what's in a genome? Yes, you can see that. What's in a genome? I've already mentioned the chromosomes, the 46 chromosomes, all of them in pairs. So 23 pairs makes up the 46 chromosomes in pretty much every cell in your body. Each genome is 3 billion bases. That's the A's and C's and G's and T's in the DNA. And times 2, you've already seen the times 2 because you saw 2 red dots, not 1. So 1 copy of 3 billion comes from mum, one copy of 3 billion comes from dad, and that's how the whole question of family inheritance works at the genetic level. You're looking at the contribution, parental contribution to what's going on. And then of course, the child's genome is a fresh genome. It's made up of parental DNA, but it's also its own genome. It has its own start. And so sometimes there are things which are unique to the child. It has more than 22,000 genes. I'm not going to say much about genes, but I'll show in various ways what genes are. These are the genes are the instructions to make proteins, which you will perhaps know a bit about because we have to eat them. But the human proteins themselves are made according to the code, and they cause functions. I'm going to show you an example of that in a minute, actually. But then, as I already said, the DNA gets misspelt, or errors happen in the DNA, and that's what causes genetic disease. Here's an example and a schematic. I'm not going to go into it much, but it illustrates perhaps something of what do we mean by these mutations in DNA. And there are very many different kinds. On the left-hand side there, you see the black is kind of normal, and then the orange is a change. So that's a change that has happened there, that orange box. It's one base in the DNA has changed, a single base change. It's a mutation, and it may cause a disease or it may not. Then there are copy number variants, and just all you have to see there is that it's a rather big gap in a chromosome. So it's not just one letter has changed, but a whole chunk, a whole sentence is missing. And so that is another kind of variant. And then there are various other ones here, very big chromosome changes that happen. And so I won't go into detail, but it just gives you the idea there are lots of different kinds of things we're looking for. And the great thing about the genome sequencing, which is the test that we're doing, is that it actually captures every single one of these variants. Many of the genetic tests do not do that. So I've said this is a new lens, a new microscope. The genome sequence on the machine that some of you saw downstairs or saw yesterday, the genome sequence is a vital way to have a look at everything at once. I'm sure you've heard of the phrase finding a needle in a haystack. It's something really very difficult to do. But that's what we're trying to do. We're trying to find the needle that causes the disease. And we've got a haystack of 3 billion bases. But the great thing is with this test, at least we have the whole haystack. So the needle's in there somewhere. That is not true of any previous test. And I'll say a little bit more about that in a moment. I'm going to tell you about Jessica. Jessica's story is an example from a very early study we did with the NHS in a project called the 100,000 Genomes Project. And Jessica's story took us from the genome, her genome, that we sequenced, to a mutation, that's the needle in the haystack, to a diagnosis of the disease, because that was the needle that we found. Is that me moving around with the microphone? It probably is, isn't it? To a treatment. And this really is the rainbow, or the pot of gold at the end of the rainbow. So this is a 4-month-old, complex symptoms, developmental delay, intractable fits. Yes, it's an epileptic case. and does not respond to any standard treatments. So problem, don't know what the cause is, don't know why, don't know what to do, what the treatment should be, and many tests have been carried out, but there was no diagnosis. So we carried out the complete genome sequence that finds the whole haystack to find every needle, to find where the needle might be. And we actually found a new mutation in the child. It was not present in the parents, which is a slight challenge, but we found a mutation in the child, and this is where it starts to show quite easily, because the example is quite easy to understand. It's a glucose transporter. So this is a gene which encodes a protein which transports sugar, glucose, into brain cells. And every cell, just like we have sugar, we need sugar to survive. Sugar gives us energy, we burn the sugar, we make the energy. But in this case, glucose cannot enter the brain cells because that transporter is missing. So the brain cells are getting starved of glucose. Every brain cell is getting starved of glucose. So that's a pretty dramatic problem with this particular needle in the haystack. But what it also gave was a clue of something very simple to do, because we know a lot about biochemistry, we know about metabolism, we know about diets. And actually we were able, I say we, I had nothing to do with this, but we were able to switch Jessica onto a high-fat diet. Because if the brain does not have any glucose, it can actually get energy from fat, just as we do. If you have a low-carb diet, you've got no carbohydrate to metabolize, so you start burning fat. And that's how hopefully we get a bit thinner. And fat degrades into ketones, and the ketones can get into the brain because there's nothing wrong with the ketone transport pathway, it's only the glucose transport pathway. So I hope that illustrates, they did reduce the seizures. It was a little late because she was already, of course, compromised by the condition she had. We couldn't completely correct the condition. But it did illustrate a totally unexpected, simple treatment that was told to us by the genome. And we had no idea what we were looking for before we started that. And that is just one example. That's what gives us all hope. A test that wasn't looking for anything. We weren't looking for GLUT1. We were looking at the entire haystack. And we suddenly saw, oh, that stands out. That shouldn't be like that. And it's only in her. It's not even her parents, let alone anyone else. And so that gives you some idea of the sort of sheer possibilities from DNA to genome to mutation to diagnosis, and ultimately potentially to a treatment that you might have known about all along, but you didn't know how to match that treatment to that person. And that, in fact, is the phrase which I was reminded about earlier. That's called precision medicine. You bring precision to medicine because you precisely know what's gone wrong, and then you can precisely work out how to put it right, which may take many more years. But that precision medicine concept has not been tried before in Ring 20. Here's one of our grand mission slides. Genomes will transform the practice of medicine. That is my thesis for much of my life now. And these are all the areas of life that it can affect. It can affect from reproduction, prenatal and newborn, screening, genetic health, which is more what we're talking about here. But also, of course, it has an impact on infectious disease, oncology, common disease, heart disease, and so on. And we talk about precise diagnosis, and I won't go into all these terms much more than that. I've already mentioned precision medicine, and that's what really matters. But the key here is, as I said to some of you earlier, an individual's genome, your genome, a person's genome, will become foundational to health. That's the haystack again, whatever needle may be in it, but we will find that needle, and the process of finding the needles will be foundational to health. But what about Ring 20? That is the key question facing us. We've never found a Ring 20 needle. And the Ring 20 itself, we'll get into why is the Ring 20, the ring itself, not the needle, and I'll get into a little bit of that later. So here's what Ring 20 looks like. You've seen these images before. Here's another genome on the left, and you see here that there's a normal chromosome 20 up in the top, but there's something rather funny down the bottom. It's the ring, and you can see in particular, I think bottom right is the best image. There's the ring right there. There's the normal 20 on the left, there's the Ring 20 on the right, and to all intents and purposes, so you've got two chromosome 20s, so what's wrong? You've got same number of chromosome 20s as anybody else, but there's something about the ring which is a bit different. Also, if you look at the images on the right, these are all the chromosomes that have just been, the images have been laid out to show the entire genome and the pairs of chromosomes. But the ring is pretty small. Doesn't comprise much of the genome, and that is what has compromised many tests so far. I mean, if you look at the image, it's a little blob until I zoom in on it. So much of the diagnoses of ring 20 are trying to look at this tiny blob amidst the sea of all the rest of the chromosomes. It's not the easiest thing in the world to pick out. Now, chromosome 20. I'm now going to examine a genome, or see how we focus in on a genome, to show you just how much information is packed in that tiny blob that the cytogeneticists can hardly see, and indeed sometimes don't see. Here's chromosome 20. It's 60 million bases of DNA. It's only 2% of the whole genome, so we're already homing in a very small part of the haystack. Over on the right is about 100,000 bases of the letters of the code. And in those 100,000 bases, this is chromosome 20, this is near the end of chromosome 20. And there are two genes which have been very interesting, I think, to people in the ring 20 research arena for some time. So these are two genes, they encode two proteins, like the glucose transporter example I gave you, but it's not glucose transporter. And these both happen to be ion channels, and they are involved in nerve regulation and nerve function, transmission of signals in nerves. And you start to see, hello, this is beginning to sound a little bit closer to home, because these are the sorts of things that happen in epilepsy. And let me assure you, I'm not giving you the answer. This is not the answer. This has been one of what they call the smoking guns, I suppose. There are genes involved in epilepsy. They're at the end of chromosome 20. It's the end that forms the ring. Surely this is beginning to tell a story. And that story so far has not proved to be the answer to ring 20. But it's still sitting there waiting to be looked at. We're looking at 100,000 bases there. I'm emphasizing these numbers because I'm going to walk you through what ring 20 has done before in a second. Let's go on zooming in. Here's one gene. Genes have exons, and in between the exons there are introns, and the exons are the bits that code for the protein. The bits in between do not, and they get chopped out. And that gene is now 14,000 bases. Now, I went and had a look at these two genes just to see what had been learned about them in the past, because it may come in handy in the future. And here's a mutation in one of those genes, ChRNA4. It is a single base change. So normally on the top row you see a C, but in the bottom row you see a T. So that's the change in the four-letter code. It changes one amino acid in the protein from serine to phenylalanine, so it's a definite change to the protein. And as a result of it, the ion channel doesn't work. And actually this does cause nocturnal frontal lobe epilepsy. So again, we're getting very, very close, this smoking gun. This clearly is the story. The story is focusing in on all the right clues to say this is how some epilepsy works. But the other thing I will say is the cause of epilepsy in that particular case was a single base out of the 3 billion. I said I was going to emphasize the numbers. So this is a slide kindly put together, I think, by the Ring 20 Consortium in the past. At least I've adapted it. I'm not going to show the whole thing, I'm afraid. But I do encourage you to look at it because it maps out the entire history of genetic testing as applied to try to find the answer to what's causing Ring 20. You start off on the left with the carrier typing, the chromosomes I've been showing you. That can get you down to about 10 million bases. You can't see anything less than 10 million bases. So hang on a minute, I've just shown you an epilepsy mutation that is only one base. So if you find a method that demands a difference of 10 million bases, I'm not going to get very far with trying to look at one base. And this illustrates the mismatch between techniques that were available in 1972 versus what's available now. And what's available now is to look at a single base, but some diseases are caused by a single base. So we've been using a sledgehammer, and I don't know what the right analogy is here quite, but the resolution of the carrier typing and testing, which is what you've all relied upon, up to and including the present day, isn't going to find every single genetic cause of every single genetic disease, because many of them are far, far smaller than the resolution. This is a very low resolution methodology. The second, which is fluorescence in situ hybridization, which again has been used, and I showed you a copy, in fact, of the red dots on the ring chromosome. That's good down to about 1 million bases. So you can see if something is there or isn't there, as long as it's 1 million bases in size. Now, on the basis of those tests, some people have said that ring 20 is not caused by any gain or loss of DNA. I don't think they can say that at this stage, and that is one of the main hypotheses for why I'm here, because my world takes us through the next 2 stages to the one at the end, which is whole genome sequencing. The microarrays are used. Not going to talk much about that, but the resolution is improved. You can see differences of 100,000 bases in there, maybe a bit less. Exome sequencing, and I just mentioned exomes in passing, the exons. You just look at the exons, you don't look at the bits in between, and you can get down to 1 base. So that might be enough. But the trouble is, with exomes, you can't see things like rings. So we're a bit stuck here. We're gaining this resolution to look for needles in the haystack, but we can't see rings anymore. We've thrown out the whole thing that we're relying on this whole diagnosis for. So there's a real problem. The only way you can see rings is with those first 2 methods. So we somehow got to bring it all together. So for my money, I don't want to be looking at just 2% of the genome, which is what the exome does. I want to look as much as possible as I can. And I've said 90% there. That's one of those scientific acknowledgements that we can't do everything yet, but the whole genome is effectively the whole genome. You can see 99% of the genes, and that's the limits of current resolution. And you can see any base change, or 10 bases, or 100 bases, or 100,000 bases, or 10 million bases. You can see all that if you sequence the genome. That's why the whole genome sequence is our new weapon, our new method of looking at diseases, just to see if we can put together these snippets of observations, the rings that people have been seeing for a long time, and the genome. And I'm very aware, I think I've alluded to it already, but I understand that the cytogenetic testing in the fish, the first 2 on the left here, the ones that have revealed ring 20, in some cases, in many cases, but not all cases, are the ones that are being discontinued in the health service. With good reason, because if we can now see a single base and 10 million from one test, why carry on with the 10 million test and not use the other one? That's a difficult transition for the NHS, but this brings me right back to the challenge that we're facing today. We can't get rid of tests if they are the only tests that test for a disease. But I'm cognizant of the fact that there are other problems that have been unanswered, which I've begun to allude to, and I'll summarize at the end, that maybe we can do better. First off, we want to be able to see the rings and why the rings, but we also want to ask the question again, so why is the ring 20 actually causing problems with genes, problems with brain cells, problems that might not require the ring itself, but something else has happened as part of the ring. And maybe for those people where we don't see a ring, maybe we just missed it, and we'll have a closer look. Or maybe there wasn't a ring, but it's still the same condition, the same symptoms that undoubtedly make it part of the ring 20 consortium, but I don't have a ring yet. So am I really part of the consortium or not? And that, of course, that's back to the question of precise diagnosis or precision medicine. We want to know absolutely everyone who has got the relevant changes that cause this disease. We might even stop calling it ring 20 if we found a lot of people who did not have a ring but were definitely in the same category, because we might suddenly find it's a particular gene that matters. So we start calling it that gene, ABCD1. We've got ABCD1. Here's the certificate. That means that this is what I'm going to expect in life. And if we know it's ABCD1, I'm jumping ahead slightly. If we know it's ABCD1, or mutations in ABCD1 that cause the disease, that cause the epilepsy, that cause the symptoms, now we know what's actually the cause of the problem. Maybe we can actually address it and put it right. And so we give this information to the pharmaceutical companies, say, can you design a drug for this? Because at the moment they have nothing to design a drug for. They can design a drug for epileptic fits, for various conditions, but it's not actually addressing the cause of the problem. So the final piece of this is framing the question or looking ahead. And I just put these together last night to summarize my thoughts as to how to shape the research project that we would like to embark upon with you and others. We know ring 20 is a chromosome abnormality, this ring. It's not a normal looking chromosome and is associated with certain symptoms. But to emphasize it again, we do not know why. That ring itself does not have a mechanism of causing neuronal dysfunction or fits or whatever. There's just no biology that suggests that. So the ring 20 is a marker, but it's not the cause itself. So we've got to understand what the ring really is and then what the cause really is. We also, as I've alluded to, and I understand, we don't always see ring 20 in patients who have the same symptoms. And this is not the easiest set of symptoms to measure. So you're in a grey area at that point, aren't you? If you haven't got a ring, am I part of the same group of diseases or not? And do I then get the same drugs that they're getting or not? There's a celebrated case, which in fact my grandmother died of. She was diagnosed with Parkinson's disease quite early, and she was put on anti-Parkinson drugs. And about a year and a half later, another neurologist looked at her symptoms and said, that's not Parkinson's. That's multiple systems atrophy, which is a related condition. And the Parkinson's drug actually makes MSA worse. So you really want to match the drug to the patient, which means you have to know exactly what the cause of the disease is to have the best chance of matching. Because if you give the wrong treatment to a person and the mechanism of the disease is completely different. My grandmother got worse, substantially worse, and unfortunately it took 18 months to pick it up, so she never really got better. She was wrongly diagnosed with Parkinson's. It happens. I'm not blaming the medical profession or anything. But we have the same problem here. Epilepsy is a complicated set of conditions, and I suspect you've all experienced different severities, perhaps different symptoms, and it's tied into all sorts of other things. But if it's in the DNA, and if there's a needle in there, that will definitively say what is the cause of the disease. And in some cases it might be one base, and in some cases it might be a million bases. But if all those different changes are affecting ABCD1, now we know the answer to the problem. We can start to figure out better what is the actual mechanism of this disease. So that's what the fourth bullet is. We need to find the molecular cause of the condition. One or more causes. It may not be a single gene, but we want to find out the multiple causes. Why does ring 20, what's ring 20 pointing to? It's not the end in itself. What's ring 20 pointing to? To find the molecular causes of the condition so that we can develop a test for it. Maybe it's just the genome. We can do that now. The machines that some of you saw, we can do this for less than £1,000, quite a lot less than £1,000, which is a lot cheaper than a whole body imaging or a scan. This is not pie in the sky stuff. This is not blue sky testing. That's why the NHS has adopted it. It's cheap and good. And we can verify the test. And I put that phrase in there because how do we know when the test is finding the cause? We can only validate or verify a test if we are able to use the test on a collection of samples of people who we know do have ring 20. Because if we say we've got a test, well, I can sequence any base in the genome. So have you looked at ring 20? No, but it's the right test. No. I've got to actually look at samples from people who have ring 20. Because until we do that, we don't really know if we can see it or not. And that is very much why I am bringing my world into your world. Because without you, the patients and the families, I cannot even begin to make some of those claims of the progress that I hope we will make. We cannot do it without genuine, real ring 20 patients. So my question slightly changed. Will a ring 20 person's genome become foundational to their health? I boldly said at the beginning, a person's genome will be foundational to their health. Now I'm not so sure. I'm asking the question, will a ring 20 person's genome actually provide the answer? I don't know, and I don't know until we join together and have a look. It may be a complete failure. So don't get your hopes up. So finding the molecular cause of the symptoms, and maybe this is getting slightly back, reaching slightly back to the first part of my talk, to the sorts of things that I'm going to be looking for. We do see gene loss in some ring 20, I say we, again, not me at all, but I just trawled the literature. There are publications out there. And yes, those 2 genes were chosen on purpose for my example, because although they simply showed how you get down to a single gene and a single base mutation and an epilepsy condition, those genes have been found to be absent. That's a sort of double negative. But you find those genes are missing in some ring 20 cases. So the ring isn't just a ring with no loss of DNA. In some cases, they've gone on to look at the ring 20 and they found that those 2 genes that are involved in epilepsies were missing. And so that begins to just suggest a connection between ring 20 and these genes on chromosome 20. And maybe, therefore, they may be more involved than we know in other cases as well. So that really is quite a smoking gun, that one. There may also be other genes involved in epilepsy that are in the same part of the chromosome. I said there were 22,000 genes on the genome. We only know the function of about 6,000 of them. We don't know what most of the genes actually code for. So there could well be some more epilepsy stuff going on either end of chromosome 20, because the 2 ends come together in the ring. We haven't really looked at the other end at all. So this is a little bit too interesting to pass up. But we mustn't let our searches be biased purely in what's previously known. That's the point of the genome. You don't just work on previous knowledge, you discover new knowledge as a result of this test. You've got the whole haystack. Let's look at the whole haystack. We'll find things that we never knew existed. And that's very important, just to take that into account, prior knowledge, but not to rely on it. Some cases have also been found to have disrupted DNA, and the ring itself is a disruption because the 2 ends have joined together. That's abnormal. But there are other DNA disruptions that have been seen in some cases. And a little memo to myself, if you remember that slide with the little orange and black boxes, inversions, where it does that, deletions, where you lose a bit, duplications, where things get doubled up, and UPD, sorry, uniparental disomy. Ask me afterwards. But I have seen it recorded as observed in chromosome 20. I will explain it. You have 2 chromosome 20s, one from dad and one from mum, and okay, one of them is now a ring. But actually, when they go and look, they do not find evidence for 2 chromosome 20s. They find the same one, which is either mum's or dad's, but not both, has now become the 2 copies. So this is mum, and this is also mum's. So dad's chromosome 20, completely arbitrary, sorry dads, but dad's chromosome is no longer part of the child. And that's what uniparental, one parent, disomy, 2 bodies, but only one parent. Uniparental disomy. So I can't resist teaching, sorry. And then we recognize we may need to look beyond the genome, because methylation, which some of you may have heard of or even may have had a test for, I don't know if methylation is used in ring 20, but sometimes methylation can silence or activate genes. So you've got the gene, DNA's okay, but it's not being switched on or it's not being switched off in the right way. And so we may have to look beyond the genes themselves to look at methylation, which is an ongoing area of technical development for us. So we need to be aware of that. As I mentioned early on, and it's in red, all testing done so far on ring 20 or many other diseases only give partial coverage. Maybe you look for certain genes, maybe you just look for rings. It's very low resolution, and we need to do better than that, because any one base might be enough to cause the actual epilepsy. And these are all amenable to new technology. So we're working, our core currency is genome. We're working quite hard on RNA, and the methylation is also there to carry on. So I think future prospects, this is the final slide, I think. I'm somewhat reiterating myself, so I'm sorry, I just sort of dumped my thoughts onto these slides last night. But establishing the cause of the condition is fundamental. It's not the ring, it's whatever's happening with the ring in some cases. We'll use the new methods that we have, and perhaps methods we're still developing. we must ensure that genetic testing evolves but does not lead to deterioration of clinical care. And that was what got me when I first heard about all this from Shirlene, and very shortly after, probably from you, Alison, as well. You know, this cannot happen. You cannot have a forward-looking, inspirational NHS. Did I just say that? that goes and introduces new technologies and calmly just forgets the fact that you're now missing stuff that you could find before. It's not good enough. We've got to improve. And certainly the people I talk to in the NHS are extremely aware of that. And we all work very hard on trying to make sure nothing is being lost as we move into the future. And we must all, I mean, not you, we must take responsibility for that, because we're developing new technology. We have to show it's fit for purpose and fitter than any previous technology if it's going to displace others. So we will resolve the above challenges, I hope. It will provide a basis for better care, not just the precise diagnosis, as I've mentioned, so you know whether you have this condition or not, ABCD1, as I've kept on calling it. It'll also open the door to other epilepsies, and that's where Samir, I think, is extremely important to this. He's not just a Ring 20 expert, he knows more about the whole spectrum of epilepsy. We've touched epilepsy before. Epilepsy, in fact, early infantile encephalopathic epilepsy, I knew I was going to get that wrong, EIEE, was actually what David Cameron's son died of. And David Cameron was the one who actually triggered our whole project with the NHS. He authorised it. He paid for it. But it was because of that very personal connection that he walked into that room you see, the one with the oval table, the cabinet room. And he looked around at all these scientists around the table and said, well, I feel a bit of a loss here because I only got a C at O level in chemistry, and that's all I know about what you do. And then he said, but in another sense, I know a lot about what you do. And then he told us all about Ivan. Ivan died, I think, about 5 years old or something, and he had EIEE. He had an epilepsy, a form of epilepsy, and died young. And so in that sense, as a parent, he knew exactly what the opportunity was. And I wanted to share that because you are all parents or with children, but you are in that same position that he was in. And he understood very much that's what we were trying to do. I do believe a Ring 20 person's genome will become foundational to their health. I do think it'll fit the pattern. We know enough about genomics to know it really makes a difference. We've solved so many diseases we never knew about in the past 5 years. I didn't have time to talk about that today. And I will once again reiterate, the then Chief Medical Officer for NHS England, who got us all going on this project, who was a great friend of mine, and who persuaded David Cameron as well, was Sally Davis, who stood up just as we were in number 10 with the final award of the grant to get started. And she stood up and said, well, I'm going to say something really important. And I thought she was going to talk about how this wonderful partnership between Illumina and genomics and the government, and we're all going to solve disease. And she just stood up and said, it starts with the patient and ends with the patient. Everything else is in between. And that is a really important sentiment, I think, for all of you to have. I've experienced it at first hand, not in my own family, but with other families. And I begin to see just how impactful all your contributions to it are, and how much uncertainty you face every day. Never mind me coming in to say we're going to help solve this problem. Obviously you encounter problems every day, and I will never know what that feels like. But I do appreciate if we can do something to help provide an answer, help pharmaceutical companies provide a treatment. There are some very clever new treatments out there which could be universally applied. Teasing, that's a teaser. Happy to talk about it, but I think I've probably gone way over time, but I'm going to stop now and just thank you all for your attention.